Good evening, ladies and gentlemen. The Institute for Mathematical Sciences, or IMS in short, is a university-level research institute at NUS. Its mission is to promote interest and research in the mathematical sciences and their applications. It brings together foreign and local mathematicians and scientists of diverse backgrounds for interaction, cross-fertilization of ideas, and collaboration in research. It also organizes public lectures to inform the general public of new discoveries, interesting ideas, and applications in mathematics, and school lectures to stimulate the students' interest in the mathematical sciences and in pursuing scientific careers. The speaker of this evening's public lecture is Professor Terry Speed of the University of California at Berkeley and of Walter and Eliza Hall Institute of Mathematical Research, or WEHI in short, of Australia. Professor Speed obtained his PhD from Monash University in 1969. He has taught at the University of Sheffield and the University of Western Australia and served as chief of the CSIRO Division of Mathematics and Statistics before he joined the University of California at Berkeley as professor in 1987. Since 1997, he divides his time equally between Berkeley and WEHI, where he is a senior research fellow. Professor Speed is well renowned for his important contributions to the application of statistics to genetics and molecular biology, and in particular to biomolecular sequence analysis, the mapping of genes in experimental crosses and treatment pedigrees, and the analysis of gene expression data. A member of the NIH Genome Studies section from 1995 to 1998, he investigated fundamental problems arising from the Human Genome Project. He's a fellow of the Australian Academy of Sciences, American Statistics Association, Institute of Mathematical Sciences, Statistics, and American Association for the Advancement of Science. He has served on the scientific advisory boards and boards of governors of a number of national and international organizations. He has also been on the editorial boards of many international journals, including the Annals of Statistics, Journal of the American Statistical Association, Statistical Science, Berkeley, and the Journal of Computational Biology. He is currently the president of the US-based Institute of Mathematical Statistics. Before I invite Professor Speed to deliver his lecture, I would like to conjecture that he's a man who never sleeps. This is through my observation that he has boundless energy and that he seems to be able to reply to emails anytime from anywhere in the world. It is now my great pleasure to invite Professor Speed to deliver his lecture on genes, disease, and genetic diseases. Terry.
Can this be heard all right at the back? Fine. Well, thank you very much, Louis, for those kind words. And of course, thank you for the invitation to speak. Thank you for coming along. Hope you're not too hungry. Maybe you had something to eat before you came. All going well? I've got to talk there. Right. I chose this topic because it encapsulates what I try to do. The worst question you could ask in the question time is, how much success have you had in this enterprise? And all I can say is, it's fun, and don't ask that question. Okay, so this the general public is something I'm rather unfamiliar with. I'm moderately familiar with mathematicians and biologists, and I'm hoping that that's principally what we have here, with an emphasis on the mathematicians. The worst fear is that there'll be people in the audience who know much more than me, and I'm just going to pick holes in my biology. Well, go ahead, just don't make too much noise about it. Okay, so this is what I'm going to try to talk about today. The major themes, which is subtitled The Story of My Life. Genes, disease, genetic disease, what we do. And there's a sort of subtext here, which is that statistics, I'm principally a statistician, despite being rebadged bioinformatics or whatever happens to get you a job. In my heart, I'm a statistician. And so I want to show why a bit of statistics comes in. But this will be very low level, both from the biology point of view and the statistics point of view. But occasionally I'll mention a little bit of typical statistics just to impress you. Okay, major themes. Whoops, there they are. I'm not very keen on stuff that comes in line by line. So, looking for genes. In some sense, you could say that's the story of my life for the last 10 years, looking for genes. Analysing many forms of data, seeking biological associations, many types, from familial to evolutionary. So, as you can see from the fact that I have highlighted certain words here in red, genes, data, and associations, they're my themes. Distinguishing real associations from apparent ones in data to find genes. So, that's the story of my life, folks. Genes, data, and associations. And I'm going to try to entertain you for the next 40-odd minutes on those three topics. Okay, genes. Well, we all know that, at least current mythology has it, that everything goes back to Mendel. There are probably a few people long dead who might feel they contributed something independent of Mendel to this story, even perhaps before Mendel and certainly after. But anyway, we'll think of Mendel as the beginning. And I'm not going to give you a brief history of the concept of gene. I'm going to jump rather quickly to some modern version, that is the so-called central dogma. What is a gene? Well, to make sense of this, I want to remind you that long after Mendel, it came to be believed that genes have something to do with DNA, and they also have something to do with proteins. And here is one version of the central dogma, which, as I was saying this afternoon to a pure mathematician, anything that is presented in biology as a universal truth is what I like to call generally true. Generally in the sense of not particularly. So this central dogma is not particularly true, but it's generally true. And that is that we have the information stored in the DNA molecule. It gets transcribed into messenger RNA, which goes off and gets translated into protein. And the bits that get translated into protein, well, we call that a gene, or if we're being picky, we call it a protein coding gene, because in some corners of the world there are people who study RNA and get really quite irritated when there is an implicit assumption that all genes are protein coding genes, and there are stacks of RNA genes around. So the DNA there in sort of plain text, as it were, some parts of it can be put into triples, and some of these triples will be translated into amino acids and through what's known as the genetic code. So one working definition of a gene, at least the protein coded one, is the bits of RNA, the bits of DNA that will be transcribed and translated into protein. It's probably good to mention, when you don't know who's in your audience, there may be some mathematicians who don't know this, or some people who are neither mathematicians or biologists, but that DNA I'm talking about is the stuff of chromosomes. So here is a sort of schematic cell, and there's a schematic nucleus with some schematically visible chromosomes, and if you sort of take them apart, unwind them, at the end of the day you get to the double helical molecule, one part of which might be a string like that. So we do need to talk about genes as things on chromosomes as well as bits of DNA. And of course it's the same thing, but it's all a matter of scale. Why does the gene continue? Well, there's far too much information here. This is just to impress upon you the simple fact that the gene is not a simple thing. The gene is kind of a complicated thing, and ever since Mendel and before, the notion of a gene has evolved a lot, and will continue to evolve. And I wouldn't dream of trying to tell you right now what is the sort of 2004 definition of a gene, how you deal with alternative transcripts and so on. But anyway, in general, the transcribed sequence is longer than the translated bit, if you remember what those words were from the previous slide. There are bits called introns and exons in general. There are bits, the codons are the triples. They have starts and they have stops. And then there are bits, which we'll be seeing in my next picture, that correspond to the boundaries between introns and exons. I guess the interesting thing from the point of view of searching for genes, in one sense of the word, is all of the things that I've mentioned here in red have statistical characterizations, which means you can look for them by searching for strings of letters with certain statistical properties. But I should also mention they're only associations. So there could be things that look just like the real thing, but are not the real thing. So figuring out what's real and what's not, that's part of the problem, which goes well beyond statistics, of sorting out real from apparent, or sorting out causation from correlation, if you like. So here's a sort of cartoon version of slightly more detail about a gene. That the picture here is, this is a string of DNA, and it's color-coded so that the fat bits, or plus size-coded, the fat bits are the exons, the thin purpley bits are the introns, yellow stuff here is outside the transcribed region, and then you can see that I'm not going to go through this in detail, but I want you at least to take away from this first couple of slides the fact that finding genes in DNA is not a trivial task. Untranslated region is what that means. The boundaries between the introns and the exons have special structure. Stop. Okay, so you can see that I've got these things highlighted, and so there's a lot of stuff I'm not telling you about, but in a sense then, if you want to find a gene, say a human gene in the human genome, and all you have is the sequence, you sort of look around for stuff like that, which in practice is not as simple as it sounds. Bits that fit together. It wouldn't be good to find an exon if you didn't have somewhere around about that exon a start and a stop codon. But the fact is they could be 50,000 or 500,000 of these letters away from the guy you're looking at, which is part of the difficulty. Anyway, a few facts just to show you that this is not a piece of cake. The genes are currently thought to be about 3% of the genome. Average length of the operating bit is maybe a few thousand base pairs. Average number of exons, maybe 5 to 6. Average exon length, pretty small. Average intron length, maybe 10 times bigger. Not too many are only one exon. Some of these exons can be ridiculously small. And here's it's not a typical gene, but it's not an atypical gene either. This guy, this is, well, I won't bother saying what it is, 17 exons, 40 to 60 base pairs long, spread over 67,000 base pairs. So this gene, if you want to look for it, you'll find a region of 67 base pairs long, and somewhere in there are 17 bits that are pretty small, and they all fit together in this more or less canonical way, but these bits in the middle are huge, and these bits, the sort of operating pieces, are very small. So hopefully you'll believe me that finding that gene by just looking at DNA would not be a simple task. Why do I say that? Because most of the 3% of the genome that we think of as genes right now are only known to be genes by computational predictions. The overwhelming majority are not biologically validated, and there may be a lot of surprises in store for us as time goes on. Okay, so I'm going to say a little more about that, but first let's get a few definitions. Locus is an old-fashioned word for a spot or a location along a chromosome. or an individual gene or a DNA sequence. So just think of locus as location, spot. An allele is an older term for a variant form or an alternative form of a gene at a locus. You can imagine if we now start thinking of a gene as some incredibly dispersed collection of bits and pieces of DNA, allele starts to become a little outdated as a concept. Sequence variation is a newer and more useful version of the concept of allele, just a difference in the letters at a locus. But you still have to say what you mean by the locus. Polymorphism, sequence variation that happens often enough to be worth thinking about from a population point of view. And finally, mutation would be sequence variation resulting in a difference that you notice. So they're just a few useful words for the rest of the other 40 minutes or 39 and a half minutes. Okay. So I have a feeling I missed a slide. No? Okay. I have a feeling I took out a slide. So I was going to tell you a little more about how you find these guys. That was going to be one of my slides where I impress you with my depth and mathematical ability. I was going to mention hidden Markov models and all sorts of fancy words that are associated with trying to find structures like that. And I've already suggested that finding them is not easy, which is true, but there are a lot of very interesting mathematical techniques currently used to do that, so that you could take millions of sequences involving millions of these A's, C's, G's and T's, feed them into the algorithms and come out with predictions. This is an exon, this is an intron, this is an exon. There's the start site, there's the stop site, wherever it is here, and so on. And get the feeling that you've found a gene. Of course, there are other evidence you will need to increase your confidence. But the algorithms that embody that structure are already pretty interesting mathematical statistical objects. But I apparently have decided not to say that. So I'm moving on. Disease. I'm going to mention 4 diseases, 3 of which I have daily association with, and one of which I wish I had daily association with, for reasons you'll see. Multiple sclerosis, I've occupied a good deal of the last 8 years on a project involving this. And I'm sure most of you have heard of it in some shape or form. If we were 100% Northern European, I'd say I'm pretty sure you have a friend or a relative who has it. The frequency in Asian populations is not so great. So it results from the progressive destruction of the myelin sheath around nerves. So this here is a whole lot of stuff called myelin, and it's a protein which is produced by cells which is not visible here, called oligodendrocytes, and basically it's sort of like insulator plastic around an electrical cable, and when it either degrades or comes apart, then you get sort of things vaguely as analogous to short circuits. Transmission of nerve impulses slowed or blocked, and that produces a whole range of symptoms which are characteristic of multiple sclerosis, being described here as MS. I say here it affects mainly Caucasians, but there's definitely multiple sclerosis in Singapore. I know, I keyed in multiple sclerosis Singapore into Google, hit return, and got lots of hits. So people are writing papers about multiple sclerosis from Singapore medical institutions, but I don't know what the rate of it is. Unfortunately, that was not easy to find out. It's certainly the most common neurodegenerative disease amongst Europeans. Average age of onset, perhaps one might call that optimistically early adulthood, or hopefully maybe we can call it late adulthood. There's quite a severe sex bias. It's chronic, that means it sticks around, and it's cumulative, which means you don't get better, you get worse. And if you're lucky, you don't get worse twice as fast as other people. And it's not terribly cheap. So in general, this is not a great disease to have. I know pretty well one of my sisters has this disease, which would be nice to think that's why I'm studying it, but nature isn't that simple. Okay, here's a second disease, which I think is probably very well known to people, type 1 or insulin-dependent diabetes, which is associated with, as it says here, the destruction of insulin-producing cells in the pancreas. And that's associated, of course, with a whole lot of problems involving sugar or glucose metabolism. Incidence is relatively high. I've got here symptoms and treatments are well known. I don't want to put a stack of stuff here for you to read, but I'm sure you're all familiar with diabetics who have to shoot up from time to time to keep things under control. So here's some reasonable looking beta cells, and here's some ones where the cells have been destroyed. The third disease, which in a sense is rather more specific, diabetic retinopathy, leading cause of blindness in the working population of the developed world, and increasing problem in the developing world. Currently, the only effective treatment of diabetic retinopathy, that means it's associated with diabetes, or if you like, aspects of glucose metabolism, not just type 1 diabetes though, but type 2 diabetes. Retinopathy means it sort of messes up your retina, and the only treatment is basically to fire a laser at your retina. Here's a laser, so you sort of fry little parts of your eye, and this actually slows down the spread of the condition, and if it's done well, it preserves the central part of the retina, which is responsible for a good deal of your vision. So this is not a great disease, and I'm pretty sure most people are familiar with somebody who, as they got older, perhaps started to exhibit the symptoms of adult onset diabetes, had associated vision problems. That is really extraordinarily common. Okay, those are the three diseases that I've been associated with. Here's the one I wish I was associated with, in a sense, though perhaps that's an ironic remark. Sickle cell anemia. These sickled cells become trapped in small blood cells. So these are sickled or distorted forms of the red blood cells that float around in your bloodstream and carry the oxygen to and from the lungs and so on. And these hemolyze readily and have decreasing survival, leading to anemia and eventually shock. So it's not good to have things floating around in the capillaries that don't actually roll smoothly. And here you see a curve. That's the sickle shape of what should be a little sort of oval, almost disc-like shaped object that has very nice fluid properties or hydrodynamic properties. This doesn't have nice hydrodynamic properties and causes some of these symptoms. So this is a disease that's been very, very long known, and why I'm introducing it is, in a sense, I'm saying I wish the stuff I was studying was so simple from the genetic point of view. But I first better talk about genetic disease. So there are my 4 diseases: multiple sclerosis, type 1 diabetes, diabetic retinopathy, which I'm involved in, and sickle cell anemia, which I'm not. Okay, so how do you tell if a disease is genetic? Well, running in the family sounds like a good idea, and that might sound pretty simple, but of course lots of things run in the family, maybe smoking or dietary habits, other behavioural traits. So we need to do a little bit more than just say, yes, my father had it, my grandfather had it, my great-grandfather had it, because that might be some nasty social habit, which has absolutely nothing to do with genes, but was just passed on in the family environment. Concordance in monozygotic compared to dizygotic twins. So they're fancy words for saying that if you have identical twins and they share whatever the trait is much more frequently than non-identical twins, then maybe it's because the identical twins have identical genes, and the non-identical twins are pretty much no closer to one another as far as genes go than brothers and sisters or siblings. So comparing concordance, so that if we said 90% of identical twins, if one has the disease, the other does, that's a very high concordance. And then you said only 15% of non-identical twins have the property that if one does, the other does. That would be quite a difference between the frequency or the concordance rate between the identical and non-identical, and you might think that's because of the genes. Now, there's a lot of stuff you could talk about ways in which the environment, in a very general sense, could differ between identical and non-identical twins. But I'm not going to say those things. We're just going to take this at its face value and look at some examples in a minute. A more general concept of concordance is what's called relative risk. lambda sub r, this is a terminology, the probability that an individual is affected given that they have a relative of a particular relationship, R, being affected, divided by the probability that they're affected unconditionally. This is a slide especially for the mathematicians and statisticians. Got some symbols here. But basically that means, say the frequency in the population in general is 1 in 1,000. But if you have a brother who has a disease, you have a chance of 1 in 100. Right? It goes from 0.1% to 1%. Then this number would be 10. Because let me say you're 10 times more likely to have this disease if you've got a sibling that has that disease. That would be a lambda sub S, a sibling, of 10. O for offspring, G for grandparent, and so on. And in general, if you wanted to really convince somebody a disease was genetic, you might compute this number. You have to do population surveys, you have to look at people that have the disease, and then look at their siblings. You have to do a little bit of careful statistics to avoid double counting or getting it biased and so on. But at the end of the day, you can estimate these guys, and then you might think, well, siblings or parent offsprings are pretty closely related, and they share about half their genome, if you think about it, which I'm not going to ask you to do yet, later. And then you go to grandparents, they share less, you can think what fraction. And then you might go to second cousin twice removed. And the idea might be that if these numbers, these relative risks, go down in some more or less sensible way as the relationship gets more and more distant, that suggests a genetic cause. Now, of course, it doesn't prove it any more than anything proves anything in this world, but it suggests it. And these ideas here are some of the evidence that you put together to convince yourself maybe genes are playing a serious role. Now, there's another point of view that genes play a role all the time, and this is all a waste of time. So, in a sense, and that's quite defensible. You could argue the environment affects everything, genes affect everything, and everything affects everything. So, what are we trying to do here? We're trying to decide whether something is affected by genes enough that it's worth looking for those genes. Right? Because everything affecting everything doesn't mean you're going to find that everything when you start looking. Okay, now of course some things are simple. A nice well-defined trait like being bald, hereditary baldness, that's the name of a particular form of hereditary baldness. You have a pedigree where the black labelling here are the individuals that have this particular form of baldness. That's a very clear-cut example of something running in the family. And I'm going to show you a simpler example of this a little bit later, and how we do a little bit of mathematics to convince ourselves there really is a story here. Okay? So, let's go to my examples. How can I convince you that multiple sclerosis is a genetic disease, that it has a genetic component that might make it worth us spending a few $1 million trying to find those genes? I'll tell you later why we might want to find those genes, but right now. So the twin concordance is 25 to 30% for identical. It's quite low for non-identical, maybe about 4 or 5%. I didn't write that. The lambda S for siblings is 25 to 40, depending on when you measure it. So that means you're maybe 40 times more likely to have multiple sclerosis if you have a sibling who has multiple sclerosis. That's me. So I don't have it, fortunately, but it's only 40 times more likely. The base rate is about 1 in 1,000, so it sort of jumps up to about 40 in 1,000 for me. But that's still not conservative, fortunately. Type 1 diabetes is not quite as heritable as multiple sclerosis, interestingly enough. But it has, well, it's much more frequent. Monozygotic twins, 30 to 70%. The sibling rate is about 15. Environment is important for both of these. I don't have time to go on about environment, but let's just say if we don't find the genes we're looking for, we'll blame the environment. Diabetic retinopathy is indirectly genetic because it's associated with both forms of diabetes, both of which have genetic components. And sickle cell anemia has been known, I believe, examples in Roman and ancient literature of referring to sickle cell anemia. So I'm not going to produce any pedigrees. It was really pretty clear cut for centuries. Okay, so Here's the sort of gold standard for people chasing genes. I just want to tell it to you. Sickle cell anemia is pretty much accepted to be wholly caused by a mutation in the beta globin gene. Now, the hemoglobin that we have inside our blood is made up of 4 little bits, 2 alphas and 2 betas. And here's the beginning of the sequence of normal beta globin. Starts off 1, 2, 3, 4, 5, 6. It's put in triples in relation to the triples that I showed you earlier. And here is the so-called Sickle variant, where this A has become a T. 1 letter change in the sequence. Now, there are actually a number of mutations, but this is the principal one, which changes the amino acid that this guy codes for, which is glutamic acid, to valine. And that might sound like a pretty harmless thing to do. And that's the spot in the 3-dimensional assembly where it happens. And it just happens that glutamic acid is what's known in the protein trade as hydrophilic. It likes water. And valine is known in the protein trade as hydrophobic. It doesn't like water. And actually replacing a hydrophilic by a hydrophobic amino acid does have consequences. And that is the sole reason why something that is a nice sort of discoidal shape ends up being this horrible elliptical shape. It forces a reconfiguration. So as I've said there, the sequence variant A is in the middle of the 6 codon. And there's a frequency of 1 to 20% in Africans, and it's now pretty small in Europeans. And this is a famous example that has been known since, depending on when you count, the late 40s or the early 50s. Okay, so why am I telling you this example about a disease I don't study? Well, in some sense, our aim, I sometimes think it's futile or excessively simplistic or naive. Our aim is to find analogues, if they exist, of the sickle cell mutation. That is, DNA sequence variants that contribute causally to the disease traits we study. That is, we would consider it an enormous success if we could say that if you change that letter to that letter in this position, you're going to have an impact on the likelihood of the individual having that DNA mutation getting diabetes or multiple sclerosis. Okay? And you might think, well, gee, why haven't they done that? Well, I'm about to give you a hint as to why they haven't done that. And they have done it to a limited extent with diabetes, and there is not a single example of a base change yet with multiple sclerosis with demonstrated causal consequences for multiple sclerosis. And that's, we are, if you like, we're meeting the group I'm working with on this at the Hall Institute and elsewhere, hoping to find these things. I say if they exist. I mean, you feel in a way they've got to exist. It is genetic. The DNA is the repository of the genetic information. So there's going to be a genetic contribution coming through families. It's got to be there in the DNA. But there are reasons which I won't try to go into now why expecting a single base change to exhibit identifiable consequences may be excessively naive. But nevertheless, that's what we're looking for right now. And you might say, why? And then I'll remind you of the famous sort of analogy of why a person looks for his keys under the lights rather than somewhere near where he lost them. And maybe you might think that's appropriate, because at least that's where we get a little bit of light at the moment. Okay, so anyway, the reason things are harder than you might hope is this simple distinction between simple and complex. The one gene, one disease story is underlies the sickle cell anemia. That beta globin is produced and goes into globin. That's not to say that's the only thing that beta globin ever does, but that seems to be pretty clear cut. Many genes interacting with each other and the environment to give a disease, that would be closer to where we are. And even to say one disease, that's already not so simple, because how do we know we're talking about the same multiple sclerosis when we look at several hundred cases? So we have problems even being sure we're dealing with the same disease, and we certainly have problems. There is tons of evidence that it's not one gene, apart from the fact that the concordance in monozygotic twins is not 100%. Right? The concordance with monozygotic twins with, to be honest, I don't know, I know that there are examples, but I'm not aware of any detail. If one monozygotic twin has the base change that gives him sickle cell anemia, and everything went fine during the process of producing those twins, the other one's going to happen. So one would expect 100% concordance from these single base mutations, unless it was a de novo mutation. We won't think about that right now, depending on when it happens and so on. Okay, so this is the world we're in. We're in we're in diseases where we are sure that it's not one gene. No, we would have no idea whether it's 5, 10 or 50, and we are sure there's an environmental influence. And I would have to justify that with more arguments and more time than I want to present now. So there are many, many examples like this. Most important psychiatric diseases, schizophrenia, bipolar disorder, other major depressions, have similar evidence demonstrating that they must have a genetic component. Monozygotic twins, pedigrees, that sort of thing. Yet it's totally clear the so-called environment, whether it's the intrauterine environment when they're being, during their gestation, whether it's the stuff they eat, the stuff they breathe, or some complicated combination of all the above, it all remains to be discovered. But environment, and in a general sense, is definitely a key component here. Okay, why are we doing all this? Well, I should have told you that perhaps at the beginning, but I wanted to sort of get started. Diagnosis. Well, it's obviously, if you had a bit of an idea of the genes whose, if you like, mutations in which may predispose you, you might be able to look at that and give some people a bit of advance warning. Counselling, same deal. Risk factor modification, variation on that. If I tell you that you are liable to get diabetes when you're 10 years old, this may possibly be something that could be acted upon. Maybe the damage was done when you were a 1-year-old. It's hopeless. We don't know. But certainly things like adult onset diabetes related to so-called lifestyle, exercise, diet, all that sort of stuff. Definitely knowing more about the genetic causes can tell you that maybe there is a chance of modifying the risk factors while you're still alive. Screening advice. Appropriate choice of therapeutics is definitely felt to be the case, and there are a few examples where treatments will be different depending on the different genetic variants that have contributed to people's diseases. Gene therapy, of course, and I would probably say this last one here, in some sense, encompasses all the previous ones, because by the time you understand the role of genes, you might be in a position to see how they work together with the environmental factors and get a better understanding of the disease, which in principle could feed back to address all these. So you don't have to think we're just doing it to get a vaccine, or we're just doing it to get a treatment. We are way, way away from that most of the time. With complex diseases, we are just trying to figure out what's going on. Okay. Right, well, in whatever time I've given myself to finish, I'm just going to say a few things about what we do to emphasise this role of mathematics and statistics. And I now realise that the slide I was looking for way back in the beginning is now about to come. So I am going to impress you still with hidden marker models. So finding genes in DNA sequence. So this is right back at the beginning where all you have are the letters. We'll be talking in a minute about finding genes on chromosomes. I say this is one of the most challenging and interesting problems in computational biology. Here are two pictures associated with two independent versions of the human genome. And now, of course, there are lots and lots of other genomes. And I've got here, with so many being sequenced, it remains important to begin by identifying the genes computationally. Now, I haven't personally worked on finding genes in the human genome, but I have in finding genes in something as exciting as the parasite that causes malaria. And to do that, we had to devise an algorithm to find the genes. Now I've got here, this is half of a generalized hidden Markov model, gene finder, for Plasmodium falciparum. That's the name of the parasite that gives us malaria. And I don't need to say what all that means, except to say that's a complicated thing, and you should be very impressed. And be aware that that is used routinely, not just in malaria but in the human genome, all the genomes that are being sequenced. The first pass effort to predict where the genes are use things that have quite a lot in common with this guy. And I know that because we copied one of them to get this guy. I mean, it has a few personal modifications. Basically, it's a variant of the most productive gene-finding algorithm with the human genome, namely GenScan, or GeneScan in its latest version. So, lots of mathematics there that I'm glossing over, but this is quite a nice way into this sort of area if you like those types of calculations. The idea is that we have things called states that represent different features. We have observations, of course. Hidden Markov model means something's hidden, something is revealed. What is hidden in the states? What is a gene? What is a promoter? What's an exon? What's an intron? The observations are just a sequence, and they embody state-dependent, composition-dependence, and other features. In a generalized hidden Markov model, if you know what an ordinary hidden Markov model is, a generalized one has duration as well. And things like reading frames, this business of the triples has to be built in, and there are two strands, and genes can be read this way or that way. So it all gets, that's why this guy's only half. There's another half down here for the reverse strand. So there's a lot of features. Remember back to that picture that had all the introns and exons and poly A and all that. You have to stick all of that into this model. And that's all I'm going to say. So that was looking for genes along DNA sequence. And I've done my share of that. And I'm going to continue to because it remains important. Looking for genes on chromosomes is a different kettle of fish entirely. Chromosome at this broad scale, where you're not worried about the individual letters, it's like saying, let's get into the ballpark. Let's not worry about trying to get the blade of grass. Just let's get into the right ballpark. And that's like the chromosome, the blade of grass you might think of as the precise place on the ballpark where whatever, let's say, the insect you're looking for is. But I'll be happy enough to get it broadly. So typically this means what's known as biotic mapping of genes in relation to mapped markers. Traditionally, markers were observable phenotypes, coat colour, blood type, serotype, enzyme subtypes. These days we have DNA markers. So you have the DNA, the chromosomes are made of DNA. You don't have to go right down to the individual letters, but you can find little places along the DNA which you can sort of go to uniquely without worrying about knowing the entire list of letters for the entire chromosome. One we use a lot are called simple sequence-length polymorphisms, and then there's a more recent sort called SNPs, single nucleotide polymorphisms. And the whole point about these markers is they have to vary from time to time amongst individuals so that they give you the information to locate them. Sorry? So here's a simple sequence-length polymorphism. This is CA repeated 9 times. And if you see there, CA, 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 I'm not going to go to 9. But you get the idea that along that, these are the 2 strands. Along this one, we have CA 9 times. There's a bit of stuff at the beginning, which is more or less arbitrary, and stuff that follows it. And this is in the middle of an entire chromosome, which might be 100 million of these letters long. But hopefully, preceding and maybe after that particular set of 9 CAs, there is enough uniqueness that you can just say, go to that spot described by the uniqueness, and there will be a string of 9 CAs. The thing is, there won't be 9 in everybody. That number will vary, and that's the thing that makes this a useful marker. The polymorphism is in that number, that length. This is how we genotype individuals. This is a very pretty slide. I'm going to take an age to tell you exactly what's there. But basically, this is a whole lot of columns of data, and each column corresponds to information it's given there in the form of an image, but when it's analysed, that image is processed as digital data, and basically down there we have these red guys, which you can see they are periodically there, size separators, and this is known as a gel, and the process that put all this stuff here is called electrophoresis, and things have been labelled with fluorescent dyes, and you can actually read off information. Each column here corresponds to what's known as a lane in the gel, has lots of information about the length, the number of repeats. If it was a CA repeat, and there's one, these different colours correspond to different repeats, so there's a big multiplexing going on here. There's a lot of data packed into that image, but it's data about lengths of sequence polymorphisms. Okay, so I'm going to just touch on what we call statistical genetics and make you think for a little bit. Large, complicated analyses of marker and trait data with specialised programs. Here's the names of a few commonly used programs: MLink, GeneHunter, MapMaker, MapMaker6, QTL. And typically what we're interested in asking is whether the trait of interest appears to be influenced by sequence variation near a particular marker. So it's all about variation, and we use, this is meant to impress the non-statisticians, we use logarithms and things like that. Okay, log-based test. So this is called a lot score, and this is one way of trying to decide whether, if you have a trait, in a way I'm going to show you in 1 second, whether there is some reason to believe there might be DNA sequence variation, maybe in a gene, near a marker, associated with your particular trait, the trait data. Okay, so here's a little sort of instructional video, you might think of it. Here's a pedigree, and in case you haven't seen pedigrees before, that's Dad and that's Mum. In fact, that's Grandpa and Grandma. And you see they had 4 kids. Woman or girl, boy, girl. And these 2 girls, later women, got married, 2 guys, and had 4 kids. And whatever this dreadful disease is, this son had it, and these 2 sons and those 2 sons had it, and women in the family are mercifully free from it. Okay, so that's our pedigree. This is going to be X-linked. I haven't got time to tell you the story about X chromosomes from scratch, but this is going to be a disease where the boys get it, and the women either don't get anything or kind of half get it, and usually not enough to notice, so they carry it. So, some X-linked trait. And I'm putting here now some information which is a cartoon version of what was in that multicoloured picture. That is little bars. I mean, if I went back, see little bars there? All those little bars are where the information is, how far they are down in the column. So here's some little bars. And you see we have 5 lengths of bars, A, B, C, D, E. And it turns out that the longer ones are near the top and the shorter ones are near the bottom. But you don't need to worry about that. So there is it. And that tells you who's got an A out of these 14 people, who's got a B, who's got C and D and so on. Okay? So at this point, if you want to do a bit of work, you can study this picture. Now I haven't really, it might be a lot easier if I put that on, because that's the same information as I have here. Person 1 up here has just an A. Person 2, that's this mother here, has a C and a D. And I've actually helped you a little by this black coating. Person 3 over here has just an E, and so on. Okay? So, what I want you to notice in this is that we have 2 sorts of information associated with everybody. We have the letters that appear to be in their blood, because this would all typically be done by a blood sample. At this particular locus. This is a particular marker at a locus. Doesn't hurt me to say what it is. It's called DSS84. So A's, B's, C's and D's are different lengths, different number of repeats. And the black coating is whether they have the disease or not. And the half black coating is something I added. Now, is there anybody under 20 who would like to say what they saw in that picture. Any interesting associations in that picture? I mean, I'll go to over 20 shortly. We'll work our way up until we get to an age where you can be expected to have it. Anyone under 20 want to conjecture? Do you see the associations in this picture? Because if nobody answers, I'm going to take you through it very slowly. Is there anyone in the room under 20? Apart from you. Can you see anything in this picture? Can you follow with me? This person is unaffected, right? Because it's not coloured dark, and he's an A. This woman is sort of half affected, and she's got a C and a D. This husband is unaffected, he's got an E. His wife is half affected, she's A and a D. This son is affected, he's a D. You started to notice anything? Are we home? I wanted somebody to tell me, preferably a young person. Mark, what have you included here? Do you see any compelling associations? Okay. Okay, so you get the idea. Now, you might ask, that could happen by chance. And of course it could happen by chance. So what we have to do is think of that as data, the phenotypic data, the data on whether they have the disease or not, and the marker data. And then we can try to convince ourselves, no, this could not happen by chance. It is very unlikely, shall we say, to happen by chance. And that's the sort of thing that log that I mentioned earlier does. So that's the sort of thing we do. And from this little pedigree with this piece of calculation, it's actually enough data to tell us, yeah, there's probably a gene relatively close to that marker that influences this trait. And that is, of course, a single gene story, and it's very nice because it's X-linked and so on. Okay, that's not what we do, unfortunately, because you can't produce pedigrees like that for multiple sclerosis. It's not quite that common. Now, of course, that's an irony. You would like it to be more common, then we would know more about it. So, in fact, our study of multiple sclerosis looks at affected individuals and unaffected individuals. And now we don't have the power of the pedigree analysis. We have to do what we call association studies. I'm not going to give any details. But here's the idea. That we think a mutation might arise or be introduced into a population. And I'm actually studying MS in a little island off the south coast of Australia called Tasmania. And it's a relatively young population. It was first populated in the early 1800s. So any multiple sclerosis that got there came on a ship. It did not arise within the Tasmanian population, at least within the European Tasmanian population, by mutation. It sailed there, and then that person had lots of children. And the idea is that a susceptibility gene, or what we call a haplotype, through time it changes, it gets inherited. Sometimes they might disappear if the person has no children, but they go through. And we try to use this information to do a study which compares affecteds and unaffecteds. We do it by getting information about these lengths, these numbers of repeats, and typically, because we have 2 chromosomes, you have 2 lengths, and we try to organise those into what we call haplotypes, which are the strings of these numbers that you got from your father and your mother. And I think that's pretty much all I'm going to say about the mechanics of it, but except to say that it's quite tricky, and well, we haven't actually succeeded in finding any genes yet, but we have some promising candidates, is what they all say. Okay, I've already said this a little bit about Tasmania. It has a relatively high incidence. One of the reasons we know, or we strongly believe, that environment has an impact is that there are some striking environmental gradients in MS. The further you go from the equator, the greater the rate of MS. And it's pretty strongly independent of the ancestry of the individuals. So it's almost certainly due to something environmental that is maybe to do with sunlight or temperature or something like that. Okay, so I've pretty much covered that. So I just want to tell you what our strategy is, to tell you, interestingly, we wrote a paper explaining the strategy for the transactions of the Singapore Medical Society about 3 years ago. So I had published in Singapore about this. We didn't have any results at the time, I might add. We're just getting them. But this is a summary of what we did, and I perhaps won't read through it. We collected all the cases. We collected the relatives of the cases. We tried to figure out what was transmitted from their parents and what wasn't. And then we looked at the sharing. What we looked for is regions of the genome that the MS cases have that the control don't have. Something that might be unique and specific to the cases. So there's a well-known concept of case control, where you look for what the cases have and what the cases don't, and then you try to decide whether that is more likely what you see than chance, because there will be differences between cases and controls by chance. And then we have three ways of looking at our statistic. We have cases versus controls, we have transmitted versus not transmitted, and we have across the regions. And this is all just about being wrapped up right now. Okay, I think that in the interest of time, I'm not going to tell you how we look at type 1 diabetes. But apart from saying that these pictures here indicate we look at SIB sharing. Again, it's rather hard to get large pedigrees with type 1 diabetes, but it's not too hard to find lots of SIBs. Lots of brother-sister or brother-brother or sister-sister, both of whom have diabetes, and we look at what they share at different things. And we have the idea of linked and not linked. And diabetes is one of these interesting things where we have loads of candidate loci. There's hardly a chromosome in the human genome that doesn't have a few places where people think there are genes predisposing to diabetes. But as of now, there are only 2 solidly validated places, not all of these ones here. So I want to get on to my final thing, which I'll just touch on very briefly. One of the places which is solid for type 1 diabetes is on chromosome 6. And there it is in red, IDD1, and that is the so-called MHC, matrix histocompatibility complex, and it's got a whole stack of genes, some of which are indicated here. And this is a little sort of lead into the final theme I want to touch on. If you look at the same region in the mouse, there are striking similarities. And I'm sure everybody's aware that the mouse is a widely used experimental organism. And it turns out that most of our studies of diabetes are in fact in mice. And what we do then is when we get some good candidate in the mouse, then you go to people to see if it carries over. And this has been done very effectively in this region. And I want to introduce you to one of my friendly mice, the so-called NOD mouse, the non-obese diabetic mouse. You want it to be non-obese, so you know it is type 1 diabetes. You don't want it to have the diabetes that goes with obesity, and that's the type 2. So I'm not going to have time to tell you how we do crosses with that, and how we do things called congenetics. It's quite a complicated process, and lots of statistics. We end up constructing mice. Now, if you look, we studied the dog mouse because it has spontaneous diabetes. But then we want to narrow it down, so you end up creating mice like this one here, which just has a little bit of the NOD mouse genome, and everything else is the standard so-called black 6 genome. And if this guy still shows symptoms of diabetes, then you're pretty sure there's something going on in that little region, because the black 6 has essentially zero incidence of diabetes spontaneously. And if you can produce a mouse, so there's a lot of mouse breeding, but there's also a lot of interesting statistics. When you get down to that narrow region, as I had here, now you've got to start looking for the genes in that region, and the circle gets complete, because I started telling you about finding genes in DNA sequence. Now we went on to finding genes on chromosomes and so on. So we've found gene in some area here. This is a large area. And then you start looking, because the genome is now sequenced. And we can look in the region that we're studying and go back to my earlier concept of finding genes. And look at all the genes that are there. And you might think this has already been done by all the people at the big centres. But in reality, when you have a very specific region you want to study, you want to redo it. Because there's still a lot of uncertainty as to where the genes are, how they're defined. It's still a somewhat moving target. So, Where are we? The region was narrowed down to a very small interval, including perhaps just one, or perhaps no genes, depending on who you believe. Well, this was written a while ago. We are now pretty sure the region we're studying has 2 genes, because the genome got updated in between the previous version of this slide and this one. And still work in progress, but it's looking promising. So then we have to start using bioinformatic tools to try to learn about the gene. We've got a couple of genes. How do we decide if they're genes that influence diabetes? That's an extra task. And we started using gene expression profiling, but I'm very conscious that I'm running out of time. And if you want to know what that means, you should be going to the workshop that is happening at the Institute of Mathematical Sciences right now. So I think with that, I'll just move on to my themes again and then shut up. So, major themes: finding genes, data, and associations. The genes are portions of the DNA and chromosomes. The diseases I'm interested in are multiple sclerosis, type 1 diabetes, and diabetic retinopathy. They are complex, and we're trying to find causal associations. That is, bits, genes that we can causally link to the trait in question. I haven't got to how we would do that with diabetes. I started to say how we might do it with, with multiple sclerosis. I started to say how we might do it with diabetes because we have a much richer understanding of diabetes and the idea of relating what we call candidate genes to it, and we also are in a position to experiment. There is no great mouse model of multiple sclerosis, so we can't do the same cross story that we did with the diabetes. Along the way, I've tried to show why and how statistics plays a role. This is a small subset of a cast of thousands, because as you can imagine, any of these long studies have literally scores of people. Particularly MS has very, very large cast. Simon Cook, my colleague at WeHide, is in charge of that. Melanie Barlow is my main statistical genetics expert. And here are some people that have helped with particular topics I've talked about along the way. In particular, Simon Corley and Andrew Worth have worked on the gene finding aspect. And people here have worked on the diabetic retinopathy. That means diabetic retinopathy consortium. With that, I'll thank you for your attention and try to do my best to answer questions, but nothing hard, please. There's nothing hard, not hard. I need a question to give me the feeling that I convey something to somebody.
Any questions? Sorry. You have one minute. What are diseases that can be new diseases? New disease. Oh, God. What can you say there? Yeah, okay. I'm going to make a brief statement. Infectious diseases are very interesting to us. I've mentioned malaria. I mean, one of the interesting things about infectious diseases, which probably is going to be more evident even in, you don't think of diabetes and multiple sclerosis as infectious diseases, but it may turn out that you will get them because of stuff they pick up in the environment. And then you'll find there's not such a huge difference between malaria and multiple sclerosis, that people's genes don't influence the disease directly, but influence their susceptibility to it. Because you get some mice that will get malaria almost, you just show them the parasite, they get malaria. And other mice, you can infect them till the cows come home and don't get malaria. So, disease is not a thing independent of the person. How you respond to a disease, whether you actually get it in the first place. So clearly there will be a story, maybe people already know about susceptibility to SARS. Right? Exposure is not enough. And of course we know that with HIV, that there are people who have had many, many opportunities to get infected in their 50s. And that's because of their genes. So, I'm just referring to this
rather depressing fact that everything is going to turn out to be influenced by everything. The problem then is, what can we work on? And we want to be a little closer to sickle cell anemia than we do to everything being affected by everything. But I'm sure emerging diseases like SARS are going to have interesting sort of interactions between the cause, the bacterium, and the host. End of the story. A question? The change, yeah, the beta S for sickle. So the ordinary beta is the standard one, and the beta with an S superscript, that's the one which will contribute to sickle. Okay.
And then when you talk about chromosome X, X has equal, ESS is equal to 1.
Say it again.
For multiple sclerosis.
Oh, yes. Oh, that wasn't multiple sclerosis, by the way. That was just a sort of hypothetical, not actually a real disease. I didn't get any details. No, the one called DS, whatever the number was, that's the name of the marker, the variation, that microsatellite marker that had the different A, B, C, D length variants. So, and then I just said gene, something near that one. We don't actually have the gene, we just have the linkage. So we just feel confident there is something affecting that excellent trait near to that marker. So the DXS some number, that's just a nomenclature for a particular marker. And that was the one that I put in the example that had A, B, C, D, E lengths. Okay, so that's a marker, not a disease gene. And in fact, I just wrote gene somewhere nearby. We don't really know what it is. In fact, that would cause the disease. And in fact, that length is already very large in terms of genes. So saying there is a gene pretty close to that marker that causes that, whatever that disease was, it's not saying too much. It's narrowing it down from the whole universe to the right country, but it's not telling you the address. Right? That's a lot of extra work, which I started to touch on with the mice. And we do that with mice by doing all this breeding and getting the nipple down, down, down. Then we go to the genome. Thanks. Thank you. One question is a whole lot better than zero. Okay, so you.
The search space and the local cortex. So the latest testing of the factor is basically the concept of the so he is at the search space
for monitoring? So the question was this. He's reminding me, reminding us that we really know very little about how many genes there are in the human or mouse genome. First it was going to be 100,000 about 5 years ago. Then it was narrowed down to maybe 30,000. And now it's expanding a bit. And the definition is getting a little flexible. And one thing I should add, the genes we find by our prediction method are not very surprisingly ones that look like ones we have already, because we're using statistical associations. And so stuff that is totally different from what we've seen before, we're not necessarily going to be able to find until we discover it by some other method. But the question was, if there are so many genes, how likely are we to find them by using these mathematical methods? And I would say, well, half full or half empty. I mean, you're going to find a lot, but you're going to miss a lot. That's what it's going to be, I think. Partly because these algorithms have no chance of finding things that are very dispersed, like the one that I mentioned, which is the Fragile X gene. That's enormous, spread over, say, nearly 100,000. So there's going to be room for lots and lots of biology to find genes, as well as using the computational methods. But having said that, the computational methods have been enormously valuable So that when we get it narrowed down to a small region, it is possible to look at that very, very carefully and say, if the whole gene is going to be in that region, this is our best prediction. But of course, we may be wrong even in that. Yeah. This is a terribly humbling activity. I don't know why these things are mathematics, where when you prove something, that's it, stop, it's right. Here, I think that. There was another question. Yeah. Well, okay. It's a good question. Unfortunately, I make that simple. You remember that picture I showed you that said there are diabetes genes sort of almost everywhere. Typically, they come from studies in different countries. So the sad fact is that it's almost certainly true. Nothing is certain, but it's almost certainly true that different combinations of genes will contribute to diseases differently in different populations. Right? So if we thought of a diabetes gene, forget it. There might be some constellation that will cause diabetes in Caucasians, and some other constellation that will cause it in Africans, and some third constellation that will cause it in Asians. And there is accumulating evidence that that is the case. That there are many genes contributing to different forms of the same disease. And that means your strategy is not going to work. His strategy was get lots of people from all over the world, very diverse. If they share anything, that ought to be a hit. And that's almost certainly true. If they did share something, that would be terrific. That's exactly what we haven't found. You know, I mean, you thought if you did a study in America and in China and in Africa, and you got 6 genes in each, and there were 2 that were in common, bang, that would be very powerful evidence. There's 0 in common. That's the problem.
And so that's why for multiple sclerosis, we're looking at the population of an island, because
Yeah,
expecting to
that's
have
right.
the same kind of multiple sclerosis.
Exactly. Yeah. For complex traits, well, people argue about this. This is sort of, but my view would be for a complex trait, keep it simple. That is, try to deal with a relatively homogeneous population. I mean, one of the problems is, what if we find something, we can't expect to find the same thing in Iceland or in Africa. So how do we prove what we got's right? Well, we've somehow got to go all the way with our population. Well, actually, it's not that bad because Tasmania is populated by Europeans, mainly British people. So we can actually go beyond Tasmania. But yeah, keep it as homogeneous as possible if you can. And that's already a catch. Go find it and find it. And if you do that, you're probably looking for specific genes, not all the genes. And that's where you hope to learn about the disease. It may be that if you found a gene in Tasmania, it won't be any use for diagnosis in most parts of the world because it's something specific to Tasmania. But the price you pay for the specificity is the greater chance of finding it. Thanks for these questions. Michael, sure.
Given the vast scope that you've outlined and the fact that different diseases have different levels of complexity of which genes take part, is it your impression that we are currently putting our research money in the right places, or are there things that are grossly underfunded?
It's a scary question. It's a variation on why are you looking for your keys under the lamp when you lost them somewhere else. It's because there's a little bit of light there, right? It's hopeless looking for keys in the dark, right? So, or a variation on that is you do what you can. And I suppose if you're a bit cynical, you would say, eventually, if it's totally fruitless, the funding agencies will start putting their money elsewhere, right? But there was a lot of success in finding genes for diseases like sickle cell anemia, so-called Mendelian single gene diseases. That's been enormously successful. But the sad fact is, it hasn't had a huge impact on human health because the major health major genetic diseases that contribute to mortality and morbidity, at least in the Western world and probably more generally, are not simple Mendelian disorders. These are typically rather rare. Now, I have to say, sickle cell anemia is not rare in Africa. And regrettably, that is not a done deal. And there are a few instances like that. But the major diseases, the heart diseases, the diabetes, and psychiatric diseases, they're all complex. We're very hopeful that we can do something about them, but we haven't got much that we can do, which is hugely better than hope to find single genes, knowing there are many. In a sense, we've taken the successful tactics for the Mendelian disorders, and we're trying to vary them a little and get to the complex ones. There is certainly not enough success to say that is a good strategy. And there's plenty of failure, but how much failure do you need before you give up? That's the question you're asking, probably. And I don't know what the answer is, but maybe the NIH or our Australian funding body, we've had 7 years funding for our MS study. We're actually going to ask for more because we reckon we're getting promising, but eventually, if it's really fruitless, it'll kind of tail off until some fundamentally new way to look at it will come. And I'm very conscious of the issue we're raising.
Well, I was more looking for opportunities, actually.
Sorry?
I was more thinking of opportunities.
Well, there's plenty of opportunities. These are hard problems. But you need some realistic prospect of success as well. And there is a little bit of success in complex traits. But most of the time is, getting back to that previous question, it's a complex trait which in a given population is actually pretty simple. In other words, there is a major gene in that population. It's not 17 genes and all sorts of regulatory syndromes and so on. But nothing really as complicated as diabetes or MS has been cracked yet. So we don't really know. One more question. 10 more questions. That'll be fine. The method of sickle gene disease is pretty much what I did in the category, right? Where you have markers and you try to get something where the marker in the population seems to track the disease. Right? You saw like that D. Everybody that was black affected had a D. And the mothers of the affected had half D and something else, right? Because they had 2 X chromosomes. The guys only have 1 X chromosome, right? That's why there was only 1 letter. So that's the strategy for simple Mendelian traits. Now, is that what you're saying? Or some other part? Yeah, I mean, of course, that gets pretty complicated if you have a pedigree of 50 or 100 people. And often it's not as clear cut as everybody who has the gene has a disease. So there's a lot of difficulties on crossing over, but that's still easy compared to old gene models. I'm not sure I've answered your question. Feel free to get it later. Do you still want to answer that? Once we've found all the disease genes, what's the prospects of genetic profile? And then I should say, well, I'm going to be wrong there. I don't even need to think about that question. I actually have no idea, because it really is such an unlikely prospect that we will know the panel of genes contributing to a complex trait anytime soon, I think. I mean, having said this, there are a few genes known to make little impact on some psychiatric disorders. Like something like my glutamic acid changing to a valin, there are some single amino acid changes like that that have a small impact on the frequency of, say, schizophrenia. There are a few examples. How solid they are is not clear. But this is so far away from really knowing the genes in schizophrenia that, of calling it genetic profiling, I think that's, I don't know. It's not a century away, it's certainly more than 20 years. I don't think I have much more than 20 years, maybe 30. So, ask a young person. Okay, take all the questions, folks.